Okay.
Should I start the introduction?
Yeah. Okay, go ahead.
Okay, great. All right, well, it's my pleasure to introduce today's speaker, Professor Sven Brandau. He's a tenured professor and group leader at the West German Cancer Center and University Hospital Essen in Germany. He has an academic background in biology from both Hamburg and Los Angeles. He completed his Ph.D. in the Bernhard Nocht Institute for Tropical Medicine and continued his postdoc work at the Research Center Borstel, where he later led the Cancer Immunotherapy Group. Since 2007, Dr. Brandau has been based in the University of Duisburg-Essen, where he continues to lead innovative research at the interface of immunology and oncology. Professor Brandau's research focuses on the immunological tumor-host interaction, with a particular emphasis on the roles of myeloid cells, especially neutrophils, which he'll talk about today, in cancer biology. His group also is advancing the development of novel immunotherapies for head and neck cancers and investigating the function of mesenchymal stromal cells in both cancer and infectious disease contexts. Beyond his research, Professor Brandau is deeply committed to scientific training and mentorship. He serves as the chair of the BIOME, the Graduate School for Biomedical Sciences and the University of Duisburg-Essen, and leads UMESCI, which is a postdoctoral training network for medical scientists. So please join me in welcoming Professor Sven Brandau. We'll turn it over to you. Thank you so much.
Yeah, Jennifer, Dimitri, all the co-organizers, Adam, thanks for having me here in this a very interesting worldwide online seminar. And you see the title already. It's today it's on neutrophils and MDSC, and there will be a focus on human, not mouse, immuno-oncology. And for the younger folks in the audience in the beginning, I also have a couple of slides on even historical or evolution perspective of the field. What I show here is a review or a paper from 2010. And you could ask, why do we still know less about neutrophils compared perhaps to macrophages or other cells in the tumor microenvironment? And this has also to do with the interest in the field. So many years back, neutrophils were not so popular, were not so much studied as compared to T cells, macrophages, dendritic cells, as you can see here. So there was a focus on the mononuclear cells. Certainly. So now neutrophils are catching up. I think more and more people become interested in it. But then the next problem for the neutrophils came, and that was single cell sequencing. So here what I show is a fairly recent study from our area, head and neck tumors, single cell, and then you look at the cell types the authors investigate, and you will not find neutrophils. And that's not because they are not there. So that has purely technical reasons. So they are there. I can assure you, and you will see later, they're also important in head and neck, but for technical reasons, they are not detectable. And the technical reason is low RNA count, RNases, fragility of the cells. And only very recently, I highlight some of the papers here by bioinformatic workflow improvement, but also by wet lab and technology improvements now. Very, very recently, neutrophils are also more amenable and accessible for the single cell sequencing, which obviously boosts the research in the field even more. And now this is the new website. And I did realize, I think this is a neutrophil up here. So I think they are now visible to everyone. So a little bit as a gimmick. So how did we get into the field? Not on purpose. We were interested in NK cells, and this is historical papers. Actually, we were interested in NK cells, looking at the mechanism in preclinical bladder cancer model, and then we looked at all different depletion cell models, and we just found by accident neutrophils are required. And this was actually one of the first, I think, descriptions at the time that neutrophils in an immunotherapy context can have anti-tumor activity. And then we also worked a lot with cancer patient materials, and then always we found this. If you separate cells over the density gradient, And in a healthy donor, you have perfectly pure mononuclear cells. But in cancer patients, we always have this. There was always a co-purification, and turned out that this is 66B positive cells. It's neutrophils. And this is not a technical artifact. This is a sign of pathological activation of cells. So what I will discuss with you today is circulating neutrophils with T-cell suppressive activity. I will discuss also T-cell suppression in the cancer tissue and more recent projects on neutrophils and nodal metastasis. So also for the younger ones, or the ones not familiar with the field of neutrophils in its entirety. So many things started here. This is a now also historical paper, 2009, at the time from Philadelphia, where with Sven Friedlander and Steven Alberta, they used TGF-beta inhibition to show that by TGF-beta inhibition, they could reprogram or switch the phenotype of neutrophils in a mouse tumor model here, and turned out that they observed a phenotype on TGF-beta inhibition, which was more anti-tumorigenic, while in the original model, it was more pro-tumorigenic. So this was not completely new at the time. So there were already a number of factors known that would drive pro-tumor activity of neutrophils. And also, I have to stress this, I think, a little bit here, that there was already anti-tumor activity known and described, and that remains true now, actually, in many of the anti-tumor cases. Neutrophil activity is shifted towards anti-tumor activity in the course of therapeutic interventions. So this is more the homeostatic chronic tumor situation, and anti-tumor in most cases is shifted by anti-tumor therapies. So, but then later it turned out relatively quickly that it's not TGF-beta alone, it's also not only interferon, it's not only one function. So it turned out that this tumor-promoting activity of neutrophils is quite diverse in terms of the function that is illustrated here. So different molecules and functions were discovered over the years to induce metastasis, pre-metastatic niche, circulating cells, metastasis of neutrophils. I will also address this today. Then the MDSC T-cell suppressive activity was in the center of the field, and also pro-angiogenic activity, direct effect on tumor cells were detected. So there was a whole spectrum of tumor-promoting activities of neutrophils. And I think this paper now sort of also pushed the field a lot, because this was one of the first very large studies, several 10,000 patients here from TCGA at the time, to look at the prognostic relevance or landscape of all the different immune cells. And I think it pushed the neutrophil field quite a bit, because it turned out that the neutrophils down here were the cell types that were among, or even the most strongly associated cell type with adverse prognosis in this analysis. So now next era were the UMAPs and the heat maps, and then the transcription heterogeneity also entered the neutrophil space. I will not talk about this a lot, but I will use some recent studies to make a couple of points if it comes to neutrophils in the tumor host. So the first point I want to make here for the introduction is, if you look at transcriptional states, in this case, there is similarities, but also quite strong differences between peripheral blood neutrophils and tumor-associated neutrophils. These are the ones that are migrating into the tumor. So there is some signatures you can find in blood and tumor, but there's also quite substantial differences highlighted here. We don't need to go into the details, as there is anyway no consensus here at this point in time, but there is also big differences. Now, when you go, in this case in lung cancer, into the organ of the tumor, the next difference is between neutrophils that actually sit within the tumor and the ones that are sitting next to it in the same organ. normal adjacent in this case. Also here, what you can see, also this is a PAN study that integrates many published papers. You see differences, certain markers, LOX1 here, VGF, activation markers, chemokine receptors are regulated between tumor infiltrating and normal adjacent tissue. So, and now the next level of granularity now within the tumor, also here within the tumor, same study, but there's many of them, similar ones. Also within the tumor, there's certain clusters, signatures that people now try to link to functions. Here in this case, it's 4 subsets, but depending on the study, you can find multiple and different ones. So this granularity certainly exists also within the ones infiltrating the tumor. So if it comes to all these transcriptional studies, I think currently where the field is or has to go to is how we can link these transcriptomic states or subsets with function, and also how can we potentially generate better even annotation algorithms that sort of give names to these different functional states. And at some point also a consensus will be probably needed on how transcriptional states are linked with function. I'm mentioning function a lot because this will be my main focus of today. Will not be transcriptomes, will actually be function, and it will be neutrophils in human tumors. And I think in this field, the MDSC concept, at least from my perspective, was quite useful, because the 2 major achievements, from my perspective, that came from the field is first that the pathological expansion and functional reprogramming has really been pioneered by this concept, how the cells acquire T-cell suppressive MDSC activity. And the second is that there was a strong emphasis that functional experimentation should go along with molecular and omics characterization. So last part of the intro here. So this is true for many myeloid cells, but for MDSC and neutrophils in particular. In the tumor host, we have the expansion phase driven by growth factors. We have normally a recruitment phase into the tumor. There is a reprogramming polarization phase by certain tumor microenvironmental or systemic factors, and then there is an effector phase of these cells. So, and many questions which were out there at the time, actually also with new technology, are not yet solved. So if you look at this, this is also almost historic, more than 10 years old. And the question here was simple, but not simple to answer. Where does this specialization and direction programming in the different now transcriptional states, but at the time functional states happens? Does it happen early? Doing the differentiation, or does it happen at the mature later stage where the environmental factors reprogram the cells? And even now, this is in the field, and many people are investigating this in the field of tumor-mediated granulopoiesis. This is not yet solved. So it was a question raised originally here in this context that I put here, but it's relevant also in other contexts and needs to be solved. So how do we see pathological expansion and activation of neutrophils in patients? We see it quite directly because I mentioned this already. So those folks in the audience that do density gradients separate blood from cancer patients, but also inflammation, infection. If you do it correctly, you have almost 100% separation of polymorphonuclear cells and mononuclear cells. Only in pathology, and then it depends on which pathology you look like, how frequent it is. Only in pathology you have this co-sedimentation of granulocytic cells with the mononuclear cells. And then it's important to mention here, there's other names for it, also LDG, low-density neutrophils. But at the time, since this is a pathological expansion and activation of cells, there was a great consensus to refer to these cells as PMN-MDSC in the human immuno-oncology setting. And it's important to mention that there's 2 subsets here. It's an immature component, and there is a more activated mature component of cells that you can that you can see here. So this I showed you already. This was the initial discovery. So you can basically then take the cells, take them also ex vivo, do suppression assays, and do find T-cell suppressive activity ex vivo. So first question at the time was based on murine data. It was proposed that a majority of this T-cell suppressive activity actually comes from immature cells. So we asked a relatively simple question at the time. So if we now go to patients and would separate the pathologically expanded and activated cells into immature and mature and intermediate cells, where would be the suppressive activity? And we can do this relatively straightforward in the human setting because there's very well-established granulopoiesis markers like 11B and 16 that we use. And then you can basically isolate double positive mature cells, intermediate cells, and immature neutrophils from the circulation of cancer patients. The immature cells you can also differentiate in vitro to show, yeah, this is really G-CSF responsive immature cells. And when we then took these cells, the result that we were getting at the time was quite straightforward, because in multiple assays of T-cell suppression and readouts, there was a clear hierarchy of the response. It was actually the mature cells, the double positive mature cells, that had the strongest T-cell suppressive activity in multiple assays. And readout proliferation and interferon gamma is what I'm showing here. So then we went on and teamed up with the Scapini and Castatella labs in Verona and looked at cancer patient and G-CSF mobilized donors for stem cell donors. And now it's important to mention, we wanted to further characterize now these primary and most suppressive cells. And as I told you just a minute ago, it's the mature cells. And I'm highlighting this because this is a difference. For example, I'm not sure whether Renato Ostuni next a couple of months will present his study on this. So in contrast to what he did, we isolated here really the mature cells and looked on a more global transcriptomic level to characterize the cells, to find commonalities and markers. And a couple of slides to this more recent study here. What we found is that there is indeed a relatively fairly large signature that is shared by these mature suppressive cells in different tumors, but also upon G-CSF mobilization. What we found, but then we were more interested to also see and identify more specific markers that would distinguish them from other cells. And when we look more specifically, and now we are looking here at the differentiation of the cells. Here is bone marrow, healthy donors. This is from the G-CSF mobilization in the blood, where you also have the differentiation stages. There were two key observations, I would say. Observation number one was that there is a certain subset, here is G1, that is normally downregulated in the bone marrow and almost shut down, but this is prolonged expressed under these granulopoiesis driving conditions, a large signature. And then there is a smaller subset of genes that were more interesting for us because it's called G2 here, and they are pretty specifically actually upregulated in these, what we call here mature ADN or mature PMN-MDSC. And we were specifically interested in finding also surface molecules that we can use for biomarker, for isolation, and for monitoring of the cells. The signature itself, as you can see here, is associated with cell activation, exocytosis, and also negative regulation of killing and cytotoxicity. So a particular marker I want to highlight here is actually CD52. So it's in this signature, it's a surface molecule, there's antibodies available. It's relatively specifically upregulated in these mature suppressive cells over the normal density mature counterparts, also over the immature component, and it's actually associated with T-cell suppressive activity. So the patients that have high expression of 52 on their PMN-MDSC have the strongest suppressive activity. So next thing this was individual centers. What we also wanted to achieve is immunomonitoring of the cells, to make it possible to look at these cells in clinical studies in a multi-center setting. And this was not easy at the time because there was little consensus on how you would gate the markers, stain them. There was also, I told you, some technical challenges in isolating the cells. So what we did is we with the support of European Union, we founded, let's say, a study group that started to look at this and develop methods, protocols, and standards to enable multi-center immunomonitoring of the cells. And I have to acknowledge a lot Nuka Cassetta, who was at the time postdoc in Jeff Pollard's lab in Scotland, who was really instrumental to run this study. So what we did is we were going through different phases, developing SOPs, eliminating technical variables that would confound the analysis and cause inter-center differences. We also standardized the data analysis and harmonized many things on the technical level. These are the diseases we actually looked at. We don't have to go into this in detail. This is the gating protocol. At the time, it was all done centralized to not have any inter-center variabilities here, and many of the protocols are available. So this is for the people doing this in the lab, a funny and maybe also interesting slide, because what we initially realized is when we did it in a multi-center study, in the healthy donors, it's always pairs, healthy patient cohort. In the centers, you can draw a hierarchy of the frequency of the PMN-MDSC in healthy conditions, from almost absent to something. And I can assure you this is not because human beings in Italy or France are different from the ones in Germany or Poland. So this is for technical reasons. So this we had to somehow improve. And now there's out a lot of protocols, how we try to standardize, improve this and eliminate technical variables. So you can look at this if you are interested in it. But this, in the end, enabled us, I think, at the time and still for the first true multi-center study where we could directly actually compare in different centers, different diseases, different cancer types, and also compare to infection and inflammation. And what we observed here is two main messages, I would say. Number one, these granulocytic PMN-MDSC are the most abundant MDSC cell type. I didn't talk about monocytic and early MDSC here, but you can read it in the paper or we can discuss. But it's very clear that the PMN-MDSC are the most frequent in cancer. And the second main message from these pan disease and pan-cancer pan-center study is that the expansion of the cells is also higher in cancer as compared to non-malignant disease, infection, and inflammation. So this was, I think, important at the time in order to enable immunomonitoring of the cells. So this we also did here in Essen and in other centers at the time. And one important message also to keep in mind, these cells are expanded once patients have cancer, but after conventional therapy, they also go back to normal. So this is shown here. So the levels of the cells here as percent of PBMC go back to normal after conventional cancer therapy. So the next important thing to tackle was, does MDSC activity exist in human cancer tissue? And then you could argue, okay, that's pretty easy to do. We just, we take the tissue, we dissociate, dissect, take the cells and do ex vivo assay. Of course, you can do it. We also do it, but there is a lot of manipulation ongoing, obviously, with the cells. So you can never be sure are the cells the same as are in vivo. And the next issue is also you certainly use the spatial context. So we wanted to keep this because we thought also the spatial context is important. So where are these cells located? So what we then developed is directly from the surgery room procedure, where we took pieces, so pieces as big as this, from the surgery, directly whole mount labeling, bleaching, ultramicroscopy, 3D image reconstruction, digital analysis, and quantification. And a few slides on the results, so this is first how it looks like. So this is this is all individual cells, and we stained for T cells, we stained for 66B cells, we stained for functional markers, and for each individual cell now we can basically quantify whether it's conjugated or not conjugated, for each T cell conjugated or not conjugated to a neutrophil, and then we can do quantitative analysis. And the bottom line of the paper is actually shown here, and I think it's quite clear cut, because what we did, we compared T cells conjugated to 66B positive cells or not conjugated. We did it for CD4 and 8, and we used Ki67 or granzyme expression as a measure of functionality. And I use this as an example here. What you can pretty clearly see is that those T cells that are not conjugated to 66B cells have higher, or the ones that are conjugated to 66B cells have lower frequency of proliferation markers and cytotoxic markers. And I think this is pretty indicative and almost the best evidence that you can get directly in the tumor tissue without perturbation that MDSC activity exists in surgery specimens of human cancer patients. If we take this to the Kaplan-Meier level, we looked in the paper in a lot of Kaplan-Meier calculations. What you see here is that even the frequency of conjugation of T cells to neutrophils is predictive in terms of prognosis as analyzed in Kaplan-Meier. We looked into a couple of functional molecules, for example, LOX1, and what you can appreciate here is actually that the strongest negative correlation of the density of intratumoral neutrophils and reduced, also negative correlation of reduced T cell effector function actually comes from arginase LOX1 double positive or MPO LOX1 positive 66B positive cells. So then one additional data slide, because there was older papers here from Evgeny, but also newer papers known on leucin and APC type HLA-DR positive neutrophils. So is there the possibility that these tumor-associated neutrophils also have a T cell stimulatory phenotype that in many cases is associated with HLA-DR expression? We also looked into this. So certainly the frequency of HLA-DR positive cells in neutrophils is much lower compared to the other ones, but they do exist. And if you actually correlate this with what I just showed you with these effector molecules in T cells, what you can see here quite obvious is that the frequency of this HLA-DR positive is actually rather positively associated with expression of functional molecules in T cells. So this sort of also is indicative of the fact that there can also be such a HLA-DR positive phenotype that is not associated with T cell suppression, but rather stimulation. So now, since we know this from patients, can we exploit this therapeutically? Now I have to go for a couple of slides to the mouse to show some preclinical data. And for the preclinical data, what we wanted to have and what we needed is a marine MDSC model. So what is a marine MDSC model? So it has, in my opinion, two features. So number one, the neutrophils have, and the Ly6G cells here, as we use Ly6G, the Ly6G cells, they have to be protumorigenic. And that's happening here. So this is isotype, and you deplete Ly6G, and then the tumor is reduced, so you have protumorigenic activity. But then also, if we talk about MDSC activity, the protumorigenic, they should depend on T cells, right? So what we also did is we depleted T cells. So you see that the tumor is under control of CD8, goes up. But then if we co-deplete anti-CD8, anti-Ly6G, no effect of Ly6G whatsoever. So this seems to be a bona fide model that meets the criteria of a marine MDSC model where protumorigenic activity is strictly dependent on the presence of CD8 cells. And also the Ly6G depletion at some point influences the frequency of T cells in the tumor. So then we wanted to use chemokine CXCR2 blockade, and in order to see whether it works, we use intravital here. So this is a subcutaneous, a ear lesion in this case, of injected tumor cells, and then you see after some time this red tomato, in this case a tomato mouse here, these neutrophils are going into the lesion. So the lesion is frequently infiltrated and quickly infiltrated by neutrophils. And this is now CXCR2 inhibition experiment, and you can clearly see they pass by. So the CXCR2 inhibition is working, and you can even see it by intravital to photon in this case, that it sort of prevents the migration into the tumor. So now if we do the experiment and do a CXCR2 blockade and combine it with the PD-1, you can see that there is additive or synergistic even activity of checkpoint of blockade of CXCR2, also works as a single agent. So this potentially can be therapeutically exploited, but the problem is, and Simon Barry, I think a couple of months ago, made an excellent presentation on clinical targeting of myeloid cells, which is not really clinically successful currently, and there's many reasons for this, for the dosing, for the timing, for the transient activity also of the blockade, compensatory mechanism, etc. So I think that currently, and we and many others, I think, are working on this. It's really crucial to even now better understand what really determines the density of tumor-associated neutrophils, including recruitment in tumors. And we are actively working on this on the mechanistic level. It's ongoing, so I cannot talk about this now. So what I can talk about now is actually in the last part of a presentation is a more recent study that also addresses one of the main functions of neutrophils in cancer that I have depicted here. So we talked about this T-cell suppressive activity quite a bit. So what about pro-metastatic activity? So if I mention pro-metastatic activity, what I mean to say is lymph node metastasis, not distant. So that not so many people work on lymph node metastasis, But it is important. Here we are in a head and neck surgery department, so 50% of the patients, they do have lymph node metastasis. It is associated with poor outcome. So it's important clinically. Also clinically, there is a neck dissection done clinically for decreasing risks of recurrence. Also to make it's used for diagnostic use for clinical decision making. So in many tumors, also the lymph node metastasis is important. There's probably less known on the mechanisms of metastasis to the lymph nodes. I'm listing some facts here in terms of the signaling. EMT is mentioned, cancer stem cell features of tumor cells that go to the lymph nodes. So I said I'm talking about lymph node metastasis, but did we want to study lymph node metastasis? Originally, we didn't. Originally, what we wanted to look at is actually microbiome, and you will see the connection in a minute. We wanted to look at the microbiome because now it started with colorectal cancer and the gut microbiome, and many of you are probably familiar with the data in the context of cancer, immunotherapy, and so forth. But it goes way beyond colorectal. So many tumors are populated, colonized by microbes. And here I'm just highlighting a couple of landmark papers in the field. So, and when we looked in our own cohort, what we realized, I mean, neutrophils are anyway, they are pretty connected to the biology of infection bacteria. So what we realized is that the neutrophil density in the tumors is actually connected to the density and colonization of the tumors with bacteria. And we were not the only ones realizing this. There's quite some evidence now from the literature. One example is here, that these bacteria, in this case, they call it immunosuppressive micro niches, where the bacteria reside, and these niches also contain 66B positive cells. 66B antigen is now familiar to you. That's the primary antigen to detect neutrophils in human cancers. So question of the study was, do bacteria modulate the crosstalk of neutrophils in cancer, and how do they do this? So in order to be able to study this, we needed a sequential system where we could do mechanistic studies, but it should mimic the crosstalk. So what we decided to do is what I'm showing here. So we use a system where we stimulate tumor cells with bacteria. Then we take the supernate and we remove all the bacteria, also add antibiotics. That's important to realize. The neutrophils are not connected to the bacteria. So the bacteria stimulate the tumor cells, and this stimulated tumor cells stimulate the neutrophils, and then we put it back to tumor cells. So in the end, biologically, we have a crosstalk, but experimentally, it's a sequential system where we can intervene and do mechanistic studies at different time points. So the first thing that we actually realized is when we do this sequential stimulation of all the neutrophils, take their supernatant, put it on tumor cells, there is clear antitumorigenic activity. Viability of the tumor cells went down. There is cell death. The proliferation rate goes down. There is certainly less cells, and there is killing ongoing. So a clear, I would say, a clear direct antitumorigenic effect. When we looked at the function of the neutrophils, how do they look like when they receive this response, bacteria, tumor to neutrophil? How do they look like? So there's increased ROS production. There is increased MMP9 activity. There's certain activation markers going up, neutrophil elastase activity. We can observe, as you can see here, we can also see these formation of nets for the ones not familiar with the neutrophil community. So these nets, neutrophil axillary traps, so that's like expelled DNA that has a primary function to capture pathogens, but it's also happening in many other activation scenarios. So also these net formation we do see in the neutrophils. So basically what we did, bacterial stimulation of tumor cells, and then the tumor cells in the next step activate the neutrophils. I showed you the net formation, the activation, MMP9 neutrophil elastase, and there was some anti-tumor activity that we could observe. So, and we did this, and what then turned out that this paper came out, which basically shows how, and also mechanistically, how neutrophil elastase can actually kill cancer cells. And that obviously, from what I told you, was kind of familiar to us. So we saw the paper and actually also figured maybe it's not even worth to go more into the details of the mechanism. Instead, what we focused on is actually what happens to the tumor cells that survive the attack. Because what I didn't tell you so far is, I did tell you some tumor cells die, but there's also a large proportion that survives. And I have not shown you how they look like, and this is how they look like. So in there, now for many of you, this is already quite reminiscent of what? Yeah, of a mesenchymal phenotype of fibroblast-like appearance. So there is less cells, but the surviving cells look like this. So then we obviously, this is very mesenchymal morphology, so we looked into mesenchymal genes by standard methods, and there were some genes upregulated, yes, but then we also did what I think is more or less a standard to do now is RNA sequencing of the surviving cells. And then it turned out that indeed, also on a more global scale, hallmarks of EMT, epithelial mesenchymal transition, anti-apoptotic genes, gene center enrichment here. So almost all pathways that contained EMT in there were enriched in our cells that responded with these EMT phenotype. So since we then have this EMT signatures, next question was on the function of the tumor cells. So what kind of function do we see? And we did pretty, I would say standard assays. If you look at aggressiveness of tumor cells, maybe not standard is this in ovo assays here that I'm showing here. So here we looked and observed increased in ovo invasion, in ovo metastasis, in ovo angiogenesis. As you can see here, all of this was pretty much enhanced in those tumor cells that survived attack, stimulated by bacteria tumor, and had this mesenchymal EMT signature. So then, to make it also clinically relevant, we went also back to an orthotopic mouse model where we are injecting tumor cells into the floor of the mouse. And then there is a certain level of lymph node metastasis in the model. It's not much, but a certain level is there. But when we then compared control and these mesenchymal EMT stimulated cells, there was a dramatic, you can see here, there was a dramatic increase in the lymph node metastasis. So these cells were really going to the lymph node in this model that normally has relatively low metastasis to the lymph node. And then the last couple of data slides and sets actually show us how does it look like in patients, because so far it was in vitro and in a mouse. So we went back to our patient cohort, and what we compared here is primary tumors from patients that have either nodal metastasis or do not have nodal metastasis. And what we observed is that in the patients that do have nodal metastasis, there was a much higher frequency of these mesenchymal phenotype tumor cells, and they were also co-localized and associated with 66B positive neutrophils, and you see quantification here. And if we separate in the patients niches of high bacterial colonization by an RNA probe up here, And look in these niches and in the areas where there's no bacteria. Also, there was quite striking difference. There was a higher frequency of these mesenchymal cells and the neutrophils also in these areas of the tumor that are colonized by bacteria. And the quantification is also here on the right. And then the last pieces of data were on the mechanism, obviously, because I already told you there is a net formation, neutrophil elastase production, MMP9. So also look, do we find these neutrophils expressing NE and MMP9 in the patients? And again, we compare primary tumors, patients with lymph node metastasis and without, and clearly there is a quite higher frequency of neutrophils expressing neutrophil elastase MMP9 in patients that have nodal metastasis. And same here again for these bacterial regions that also feature these neutrophils that do have neutrophil elastase and MMP9. And these neutrophil elastase is also important mechanistically because in the last mechanistic experiments, we tested the sensitivity of these mesenchymal metastatic tumor cells to NK attack. First in vitro. In vitro, what we saw is reduced NK cell cytotoxicity, reduced conjugation with NK cells of these mesenchymal cells compared to control. And then when we use inhibitors, DNase degrades DNA to eradicate the nets, or civilistat as a neutrophil elastase inhibitor, this sort of reverts the situation. And the last data set here is on the mouse model. I already showed you this, the metastasis, and what you can see here is the metastasis is indeed under the control of NK cells, because if we deplete NK cells, metastasis goes dramatically up, so that this altogether suggests that net formation, also MMP9, increases tumor cell resistance to NK killing, and this enables metastasis to the lymph nodes. So that I would summarize this part here with saying that according to the conventional scheme in the neutrophil and cancer field, there is different functions, phenotypes of neutrophils that can be categorized or have anti-tumor activity on the one hand, but can also have pro-tumor activity depending on the neutrophils that you look at the conditions. What I would actually propose is based on the data that I just presented is a little bit an alternative model for this dual role, because it can also happen, or the decision can also be taken at the level of the target cell response. Because what I showed you is that there is anti-tumor function of the neutrophil population, and a certain proportion of the tumor cell population was indeed killed. So this is clearly anti-tumor activity, but there were cells that were resistant and not killed, and they underwent this phenotypic switch. And this switch was quite pro-tumorigenic, because the EMT induction was associated with lymph node metastasis and with escape from NK control. So the decision is also taken at the level of the tumor target cell response. So the overall summary for the presentation for today would be from my end that the MDSC activity is in the human setting of circulating cells is mainly executed by mature cells. It does exist in the circulation, but also in solid tumor tissue, and it also has clinical relevance. The appearance of these pathologically expanded activated cells is clear in cancer and disease, but the cells are virtually absent in healthy donors. We identified certain intertumoral niches that are colonized by bacteria. These niches recruit tumor-associated neutrophils, and this is then leading to induction of EMT, progression to lymph node metastasis, and escape, innate immune escape, I would say, from NK cells. And then in terms of the pro and anti-tumor activity that is pretty well documented in the field. It can not only be regulated perhaps at the level of the neutrophil, but can also be regulated at the level of the target cell, because some cells can be sensitive and killed, and others can be reprogrammed and resisted and metastatic. And this is the acknowledgement of the funding agencies and the collaborators. And yeah, I thank everyone for the attention, and obviously I'm happy to take questions if there are. Thank you.
Thank you. It was wonderful. Thank you very much. It looks like there is no questions in chat yet, but people probably will start. I will give you some questions. Two small questions. Number one, in your part with the bacteria, did you try to figure out the relationship between your experiment in vitro with bacteria having very exciting effect and real presence of bacteria in tumors and neutrophils? Basically, to put it simply, where the neutrophils find so much bacteria to be activated with in cancer, and whether this not some kind of extension of artificial experimental model into the patients. It's first question.
Yeah. Good. I mean, what we cannot do is a functional in vivo. That's obvious. So what we did find is that we have a number of bacterial probes that actually do detect quite substantial levels of bacterial colonization, given all the limitations of the technology. Yes. But there are certainly areas that are quite substantially populated by bacteria, and as I showed you, also the phenotype of the tumor cells and also the phenotype of the neutrophils is different in these regions. Yeah, so which sort of tells us at least that the biology that we saw in vitro is somehow reflected in vivo, and also that shows us that these areas of the tumor that are colonized by bacteria in terms of neutrophil phenotype and tumor phenotype differ from the rest. And
okay, you have a second short question, also more fundamental nature. So mature neutrophils, which is a very important piece of data, they are highly immunosuppressive. So then, how do you envision those mature neutrophils inside the tumor will be repolarized after interaction with tumor and then go there? Because the nature of neutrophils is really, it's like a sack of enzymes and activity in the mature state, living only a day or two in tumor, maybe 3 max. And how do you envision that they would be repolarized in situ, in the tumor?
Yeah. I mean, the issue of how long they live in the tumor, I still think there's some controversy on this in terms of the exact timing. Certainly they live longer. Also, you mentioned live up to 3 days. I would not exclude actually in human full tumor microenvironment setting, if you take, for example, ex vivo explants, you can see them quite long. So I think they live substantially longer than ex vivo. So I think this is very likely, although we cannot experimentally test it, but it's very likely. So they live much longer. So which gives them, first of all, that gives them some time also for the transcriptional machinery in reprogramming. So I think it could potentially happen because of this longer lifespan. It's difficult to do this in vitro because I'm pretty convinced that they live much shorter in vitro. So there's some, I think, possibility for transcriptional reprogramming, but it can also be some level of differential activation. So depending on which stimulus they receive, and of course they have an arsenal, but the arsenal is quite big. I mean, they have preformed in granules multiple molecules. that they could activate, also secrete. So it's transcription, it's secretion, and it's degranulation. So I think there is, I envision, if you ask me how I envision, I think there is quite some potential over the time that depending on the signals they receive, that there is a differential response on transcription, secretion, and also differential, I would call it differential degranulation, maybe.
I have a question. Several questions, but you focus on showing the different populations in blood and then those in the tumor and the mature neutrophils. Do they have a consequence in the tumor, and do you ever study them? Like, do they get into the tumor? And can you distinguish them from the PMN-MDSCs that do the mature type that get into the tumor?
Yeah. So the question is whether these are pathologically expanded cells we see in the circulation, whether we studied whether they go into the tumor and whether we can find them in the tumor. Yeah. I think what happens is, what we can say is, we not completely studied it to the very end, but to the data that we have currently, what we can say is the immature cells.
Mm-hmm.
Immature cells are actually less migratory. So there's an intrinsic feature that they have. So if we function experiments in terms of, we provide chemotactic factors, tumor condition medium, factors from the tumor microenvironment. If we provide these factors, the immature pathologically expanded would be less responsive, which would be, if you convert this to the patient, would be indicative of that they are not very actively recruited. The more mature ones, that also suppressive ones, are more actively recruited. So this is what we can say for the relationship circulation versus tumor. So, and then in the tumor, obviously, we and many others, we see, depending on spatial arrangement, we see different phenotypes appearing. And I showed some of the phenotypes.
And one other quick question. A lot of us focus on a single cell type like neutrophils or macrophages. Have you ever seen any interaction between the PMN and MDSCs and macrophages, like physical or signals or correlations in these settings that you study?
Yeah, that's a good question, and yeah, we are also looking into this a little bit, and it's very difficult to have conclusive answer on this, but it could well be. Yeah, it could well be. Well, let's put it this way. Some of the data point in the direction that also neutrophil macrophage interaction is or executes some of the neutrophil. Some of neutrophil activity, and it's also in depletion experiments, the macrophage are affected, and there's also areas of co-localization activity. So could also. There is probably a neutrophil macrophage T cell triangle.
Very interesting. Okay, thank you. Let's see, who would like to manage the questions? Dimitri?
Yeah, I can do it, but Jennifer, okay, I can do it. That's fine.
No, I'm happy.
I can read it. It's okay. Do the pro versus anti-tumor function correlate with tumor stage?
Do the pro and anti-tumor function correlate with tumor stage? Okay, pro and anti-tumor function at which level? So let's interpret it in a way that we look at the circulation. First, there is a certain correlation of these pathological expansion, with tumor stage, but it's not absolutely striking in terms of the mature ratio or the absolute frequency. But certainly a bigger tumor, so higher stage, I mean, there is stronger signals, and there is stronger pathologic expansion. So this happens. And the pro-tumor function in terms of the intratumoral, okay, therefore from our data, we have these more T-cell suppressive, whether T-cell stimulatory, and whether this correlates with tumor stage. I must say, I'm not even sure that we sort of analyzed the data, that we separated this pro and anti versus an early versus late tumor stage. This we didn't do. I don't think we do it. One piece of data that I want to highlight here, if it comes to tumor stage, maybe that's also background of the question. In many publications nowadays, the notion is sort of transported. Early tumor stage, neutrophils are more anti-tumor. Late tumor stage, neutrophils are more pro-tumor. We actually addressed this clinically. So what we did is we looked at Kaplan-Meier curves, prognostic value of neutrophils in early versus late stage, because this is what you want to see or expect, yeah, that there's a difference of the Kaplan curves, prognostic early versus late stage. But this is not the case. So in our cohorts, there is no, so I would put it this way, our cohorts, do not support the interpretation of the data that neutrophils are anti-tumorigenic in early and pro-tumorigenic in late stage. I think there's a lot more needs to be done also with clinical relevant data sets to come to this final conclusion.
So your data indicate that in early stages, they also associate with a bad outcome?
Yeah, so we have, well, we looked at different tumor cohorts, yeah, and in the different tumor cohorts, it's not as I tried to indicate. In many cases, also in early stage, yeah, also in early stage, they are associated bad prognosis. And there are certain cohorts where in later stage, good prognosis. So this is really not consistent with the idea of a differential function in early versus late stage, clinically. Yeah.
In your tumor tissues, when you look at neutrophils versus T cells, you have the data. Did you notice the interaction in numbers? Like, for instance, neutrophil-enriched tumor part has less T cells in it, and vice versa. That's what when I look at your figures, you didn't talk that much about it, but when in the paper, but when I look at pictures, it looks like you have area of neutrophil infiltration, and the T cells are not there; they're actually somewhere outside. And is my interpretation correct?
Yeah, I think there's two important elements: neutrophil T cell interaction. The interpretation is correct. Is correct in a way that there is in many patients there is actually inverse correlation. It's correct. So more neutrophils, less T cells. It's also correct in a way that certain areas of the tumors are populated by neutrophils and then there is no T cells, and certain areas of the tumor where there is T cells and no neutrophils. So, so these areas exist. It's correct.
Great. Let me get back to the questions because there's several. Your talk raised a lot of questions, so I would like to ask every one of them. Do we think that temporary versus pro-tumoral neutrophils can arise similarly post-radiation therapy?
Temporarily pro-tumoral,
basically, tumor can arise after radiation, right? Yes.
After irradiation. Okay. The problem is post-irradiation. I cannot say anything on tissue post-irradiation because we don't have material.
Okay.
Post-radiation to look at. The only thing that I can say is in the circulation, curative radiation therapy also normalizes and brings down these pathologically expanded cells to almost normal level.
Okay. And the next question has an answer in it, but if you agree, just say yes or no, and we move to another one. So the question is, do you think some of the challenges with targeting neutrophils clinically may be related to a lack of biomarker to enrich for patients with neutrophil-dominant tumor microenvironment?
Biomarkers to enrich patients? Well, we can. Well, I mean, we can. No, it's not. We can easily enrich for patients with neutrophil-dominant microenvironments. I can actually just by our own data from our patients, I can separate the survival curves for prognosis very, very much. I mean, I can generate curves that stay up and go down quickly just with 5 markers on neutrophils and T cells. So it's highly prognostic. So we can, of course, we can enrich.
Right, that's what the nature of the question, if I understand that question correctly, is that why we don't use those particular markers, biomarker, to select patients for treatment. That's what basically that's suggesting.
Yes, yeah, that's not done. Yeah, that's probably that's not done, and certainly this would be enrich the likelihood for positive results. Clear.
Another question: did you measure any direct effects of neutrophils on NK cells in the experiment with bacteria-conditioned tumor cells?
In the setting I presented, we on purpose actually we didn't look at it. Direct effect of neutrophils on NK cells. There we didn't look in this bacterial setting. In another project we did, but unfortunately I cannot recall the exact experimental.
Good.
No.
We are finished with the questions in the chat. Maybe someone wants to ask the question or turn it on and ask. It looks like Shweta wants to ask question, right?
Yeah, it's very great.
Go ahead.
Yeah, thank you, Dimitri. Yeah, so my lab works on PMN-MDSCs in pediatric tumors, neuroblastoma, and we are currently evaluating patient samples. So my question is that, have you seen very high expression of LOX1 in PMN-MDSCs, and can you use it as a marker to differentiate PMN-MDSCs and neutrophils in these tumors, which you have evaluated?
Yes, so for the LOX1, it's clearly upregulated in the tissue infiltrating over the circulating cells.
Oh, okay.
So it's much higher in the tumor associated. Within the tumor associated, there is a higher suppressive index, as I presented it, for the LOX1 positive over the LOX1 negative. In the circulation, this correlation, we do not find this correlation. So if you ask me directly for LOX as a proxy or as an indicator of suppressive activity from our data, I would say yes for the tumor infiltrating, but not for the circulating. So that's our data.
Okay, thank you.
It's another question, please. Xin Liu, right?
Yeah, Xin. Hi, Dimitri.
Go ahead. Okay, go ahead.
Yeah, so wonderful talk. Thank you so much. I have 2 questions. My lab at Notre Dame, we study MDSCs and neutrophils as well. So first question is, so can you comment on the feasibility and also maybe actually the status of using those cell surface markers you and others have identified, such as LOX1, CD84, or CD52, as a therapeutic target to target those MDSCs? The second question is, when you have these bacterial experiments, or maybe in vivo, this also can happen, why don't the neutrophil just go ahead and eat them, the neutrophil, like what they typically should do?
Okay, so for the therapeutic target, I mean, we haven't done it so far for the new markers. I mean, obviously, we would be happy if people try it because the study, it's relatively recent study, the one presented with the 52, for example. So it's possible. I mean, let's put it this way. The aim of the study was to find the best possible surface marker for monitoring and maybe also for targeting. And this is what we came up with. So, but so far, we ourselves didn't do anything into the direction of using as a targeting molecule. But if people would like to go into it, I think these are good candidates, certainly. I mean, because this was the purpose of the study, also to identify those markers. And in terms of bacteria and neutrophil in colonized tumors, Well, I think probably neutrophils go there, and probably they also eat them. Yeah, it will happen. But certainly, what's also clear, otherwise we wouldn't find them. They don't eat all of them. This is also very clear. So they will eat them, but they will not eat all of them. Also, what is a hypothesis that we are following is that the neutrophil function, phenotype, and even also antibacterial capacity is perhaps different, yeah, in different regions. So the capacity to eat. So this is all possibilities in follow-up studies that we are looking into. I mean, there is a very complex. I mean, we talk about bacteria, tumor cells, mesenchymal cells. It's a multi-directional thing. What we have is a unidirectional, so we were able to look at mechanisms there, but I cannot exclude they will eat them. Last part to answer the question, what I can exclude, I didn't talk about it too much. What we did stimulate neutrophils directly in vitro with bacterial compounds to see whether they induce EMT phenotype and nodal metastasis. They don't. So bacterially stimulated neutrophils don't do the same thing as the ones that are stimulated by tumor cells via bacterial stimulation. Different mechanism. So this we can say.
Thank you very much.
Okay. All right. I don't see any questions anymore. People who wants to ask them. It looks like going once, going twice. Solved. So Sven, thank you very much indeed. It was fantastic presentation, very interesting. I hope we will see it again soon.
Yeah.
Thank you everyone for the participating, and see you next month, the next meeting.
Okay.
Thanks, Sven.
Thanks
Thank you,
everyone
everybody.
for joining.
Thank
Also,
you.
also from my end. Thank you.
Bye-bye.
Bye-bye.